we continue our lecture on amino acid titrations. We spoke about certain things in the last lecture where we discussed the acid base properties of amino acids, the ionizable groups that are present, amino acid titrations and PI determination. But we considered only the basic characteristics. So, if we look at amino acid characteristics, we see that we have this specific structure for the amino acid where we have what is called the alpha carbon, a hydrogen, an amino group and a carboxyl group attached, which is common to all amino acids. The difference we find is in the R group that actually determines the hydrophobicity, the size, the charge, the secondary structure, preference, the aromaticity and other special characteristics which we will come to as we study more about proteins. The amino acids we know are amphoteric in nature. What does this mean? It has an acidic and basic property to it. It has specific pKa values and these features influence the acidity and the basicity of the specific amino acids that we talk about. Now, if we go and try and understand what the isoelectric point of an amino acid is, we learned in the last class that we have something called the Henderson-Hasselbalch equation and we learned how to derive it based on the dissociation of acids, and we can find what is called the isoelectric point, which is the pH at which the molecule does not carry any net electric charge. And using the Henderson-Hasselbalch equation, in the last lecture we learned how to derive a quantity called the PI, the isoelectric point, from a knowledge of the pK1 and the pK2, which for amino acids we would consider the pK1 to be the pK of the alpha carboxylic group, that is this group where all of it is protonated, but this is also an H, where we have pK2, that is the pK of the alpha amino group, positive charge here, where we consider this group. This is what is called the zwitterionic form of the amino acid. Now, if we look at the acid base properties of the amino acid, we have a series of amino acids listed here. We have the alpha COH values. You can see the pK1s are very low because this is a carboxylic acid. On the other hand, the amino group has relatively high pKa values, all the pK2 values. Interestingly, we do not have any pKr value here. As soon as we come to amino acids with pKr values, we will see how our titration curves will change. For the example that we have here and we discussed in the previous lecture, we are looking at glycine, the titration of glycine that has a pK1 for the loss of this hydrogen, this proton from the carboxylic group. The next pK2 value is the loss of this hydrogen from the amino group, rendering this to be a deprotonated form, complete form, where this corresponds to the pK2, this corresponds to the pK1. And the PI can be obtained by using this relation where we have pK1 plus pK2 by 2. So, the structure that we saw in the first case where we have a glycine that is positive, where we just have a positive charge, pK1 means the loss of this proton, so it renders this neutral. Then we have pK2, which is the loss of this proton, rendering this as the Gly negative. So, from the pK values we can calculate the PI, that is the isoelectric point where the amino acid is neutral. This is the neutral form of this amino acid. So, the PI becomes the average, and with the knowledge of the pK1 and the pK2 values, we determine that the PI for glycine is approximately 6. This experiment will be demonstrated to you when the experimental part is shown. So, if we look at general rules for amino acid ionization, we have the alpha carboxylic acids that ionize at acidic pH, and they have pKa values that are less than 6, and where we are looking at, if we look at a fully protonated amino acids, the alpha carboxylic acid will be lost first. The proton belonging to the alpha carboxylic acid will be lost first. Then if we do not have an ionizable group present in the R, that is the R side chain, then what is going to happen? The amino group is going to finally get the proton associated with the amino group is going to dissociate, and we are going to have then a negatively charged protein, amino acid. Now, most of the 20 amino acids are similar to glycine, just like the table I showed you, where we have a pK1 and a pK2 value, and we can determine the PI value, which is just going to be the average of the pK1 and the pK2. However, there are specific exceptions to these. We will look at these exceptions where we have the R group, that is the side chain, that is also ionizable. So, if we have a PI of a compound with more than 2 pKa values, how do we determine the PI value? The first thing is we have to find the amino acid form with no net charge, that is a total charge of 0. We take the pK of the amino acid form going towards +1, the +1 form as the lower pK value, and then the amino acid form going to the -1 form, and we average these two pKs. This will be demonstrated to you in the following slides. So, what are we looking for? We are looking for the charges on the amino acid residue, but we have to remember that we do not have only the pK1 and the pK2 anymore. In addition, we have what is called a pKr, which is due to the side chain. So, this is the pK1 value of these amino acids, the pK2 values of the amino acids, and in addition we have the pKr groups. We are mostly concerned with these 5 amino acid residues because we will see how they affect, or their effect is actually more prominent or more important in their presence in proteins. So, what do we have as the 5 complex amino acids, if we call them? We have glutamic acid, aspartic acid, lysine, arginine, histidine. So, what do these amino acids have in common? They have, these 5 amino acids have 3 ionizable groups. The common part for each amino acid is the amino group, where it has a proton, the carboxylic group, where it has the proton. In addition, we are not only going to look at the pK1 and the pK2, but we are also going to have a pKr. So, we have 3 pK values. What are we looking for? We are looking as to how to calculate the PI value of the amino acid. So, we have a PI that is going to be the average of the pK below, that is the -1, the pK above, that is the +1. So, we are looking at the charge of the amino acid between AA +1 and amino acid 0. So, there is a pK that is going to be responsible for the charge going from +1 to 0. Similarly, there is going to be a pK responsible for the charge going from amino acid charge 0 to amino acid charge -1. It is these 2 pKa values that we have to find out, and the average of that is going to be the PI of the particular amino acid. For example, let us look at the charged amino acids that we have here. These are the acidic amino acids. We have aspartic acid, we have glutamic acid. Now, if this is the completely protonated form, what do we mean by that? We mean by, when we are looking at the amino acid, what do we have to observe? The alpha carbon, the amino part, the carboxylic acid part, and in this case what is blocked in yellow here is the R group. This R group belongs to aspartic acid. The 3-letter code is ASP and the 1-letter code is D. When we look at glutamic acid, we see that if we look at the top portion here, it is identical because these are all amino acids. But what do they differ in? They differ in their R group. This R group has an additional CH2 associated, which is glutamic acid, 3-letter code GLU and 1-letter code E. So, when we look at these 2 amino acids, we see that there are specific pKa values associated with the protons. So, what is going to happen when we do a titration? For example, if we look at aspartic acid, as we do a titration, the first proton that is going to be lost is the one with the lowest pKa value. This corresponds to the carboxylic acid group of the amino acid, not of the side chain. As we continue, the next pK, the next lowest pK is the one that corresponds to the side chain of aspartic acid, that is the pKr value. So, it is the pK1 value, the pKr value, and then as we continue the titration, it will be the pK2 value. Similarly, when we consider it here, it will be the pK1 value, the pKr value and the pK2 value. So, this is how we would consider it for acidic amino acids. For basic amino acids, we would look at the, so we have again the same amino acid characteristic, the amino part, the carboxylic part, the amino part, carboxylate part for lysine, for arginine, for histidine. Again we have the 3-letter and the 1-letter codes given here, and these are the specific side chains. What we observe here in case of lysine and arginine is that the protons that are going to be lost are lost in a different order. The first proton to be lost is going to be the carboxylic acid one. The next proton, if we look at the pK values now of the side chain and the amino group of the amino acid connected to the alpha carbon, then we see that this value is lower, which means that this proton is going to be lost before this proton. The same with arginine. It is this proton that is going to be lost before this proton is lost. If we come to histidine, we see that the first proton to be lost is COH, that the carboxylic acid, then is the side chain proton, then is the pK2. So, we have a pK1, pK2, pKr, pK1, pK2, pKr, pK1, pK2, pKr, and depending upon their values, you will have the loss of the protons. Let us look at aspartic acid. When we look at the first completely protonated form, what we observe is that there is a side chain. So, this is the alpha carbon where we have associated the amino group, the carboxylic group, and this is the CH2 COOH. So, what is this? This is the completely protonated form. The pK1 corresponds to the loss of this proton. So, this renders this COO minus. Then we have pKr because this is the next one. So, this is what renders this minus. Now, let us look at the charges. What do we have here? The total charge here is +1. The charge here is 0. The charge here is -1. And the charge here, the pK2 corresponds to what? It is going to correspond to the loss of this proton, which means that now we have NH2 and the charge here is -2. What we learnt that in determining the PI, we have to look for the amino acid form that is +1 to 0, the pK responsible for going from +1 to 0 and the pK value responsible from going from 0 to -1. These are the values that we have to consider. So, if we look at the 4 forms of the structure that we have here, we see that this is the 0 form. What does this mean? It means we have to take this pK value and this pK value, the average of those values is going to be the PI of aspartic acid. This is the titration. So, we have a pK1 that is close to 2, we have a pKr close to 3.9 and a pK2 9.8. So, what is the charge going from pK1? It is going from +1 to 0. For pKr, it is going from 0 to -1, and here it goes from -1 to -2. But we are interested to determine the PI from this set. So, it is pK1 plus pKr divided by 2. In case of lysine, what are we going to see? We are going to look at the charges again. In this case, we have the alpha carbon atom here, the amino group, the carboxylic group and the side chain, which is CH2, 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 NH3 plus. Now, when we look at this, we see that this charge now at the present stage is +2. The pK1 we know corresponds to the loss of the proton of the carboxylic acid group. So, we have a CO minus. The charge here now is +1. The loss of pK2 now we remember that it is going to be the amino group of attached to the alpha carbon. So, this renders this NH3 plus to NH2. The charge now is 0. When we go to pKr now, the loss of this proton, the side chain proton, is going to make this -1. What are we interested in? We are interested in charge changes from +1 to 0 to -1. So, what is the charge here? +1. What is the charge here? 0. What is the charge here? -1. What are the two pK values responsible for this? pK2, pKr. So, what will be the PI of lysine? It will be pK2 plus pKr divided by 2. So, that is how we will be able to determine the PI values of these specific amino acids. We look at the titration of histidine now. In case of histidine, again we have 3 ionizing groups. It is an ionizable amino acid. And the most interesting point about histidine is that the pKr value is close to 6. And this can vary depending upon where histidine is positioned in the protein. And because it can vary from around 5.8 to more than 7.5, it is extremely important in physiological processes and as enzymes in proteins, where we see a lot of histidine residues present at the catalytic site of proteins. We have the 3 ionizing groups. What are these? The alpha carboxylic acid, the side chain amino acid and the alpha carbon. Now, if we look at the charges again, these are the structures of the histidine molecules. When we have the pK1 value, we know that we have 2 positive charges being a basic amino acid. So, the total charge here is +2. The pK1 again renders the loss of the carboxylic acid proton, making this a charge of +1. We come to a pKr value here, where we are now rendering this as a 0 charge, because we are now losing the proton on the side chain. Then we go to the pK2 value and we look and see that the loss is going to now be the amino group attached to the alpha carbon. So, we have now NH3 plus going to NH2. So, what are the charges? The charges are +2, +1, 0, -1. What are we interested in? We are interested at the specific point where the charge is going from +1 through 0 to -1 and the pK values responsible for that. So, what are the pK values responsible for it? It is the pKr, which is 6, the pK2, which is 9.2. The average of that gives us the PI of histidine. We have here the histidine titration curve. So, based on these types of titration curves, based on an understanding of the titration curves and what forms of the molecule are present, we can actually determine the PI values of the amino acid residues. So, we can consider the PI of a normal, what do we mean by a normal amino acid? One that does not have any ionizable group. In this case, we would call it normal because it would have a pK1 and a pK2 without a pKr associated with it. However, when we consider the ionizable ones, the specific ones, the acidic ones and the basic ones, it is important to remember that the pKr values also are extremely important, and there is a way in which we can determine the PI value of an amino acid that has an ionizable side chain. This will also be demonstrated to you in the laboratory. The next thing that we are going to consider now, what happens if we have two amino acids linked to one another? If I have a dipeptide, is it possible to determine the PI value the way we consider it for a single amino acid? And the answer is yes. Now, how do we? First of all, we will be understanding more about peptide bonds when we consider a little bit about protein structure to understand what we mean by protein denaturation in subsequent lectures. When we are talking about a peptide bond, we have one amino acid that has the R1 side chain, another amino acid that is the R2 side chain. This is written or demonstrated or displayed in a very simplistic manner, and what we say is, in a condensation reaction, what happens is these two connect to form what is a peptide bond. And now we have a dipeptide, so we have an NH3 plus, a COO minus, and this CONH does not have any charge associated with it. This is the peptide bond. So, if we were to determine the PI of this particular dipeptide, what is the information that I need? The information that I first need is the pK2 value of the first amino acid, because we are looking at the NH3 plus, the pK1 value of the second amino acid, because we are looking at the COOH, because if we consider a complete protonated form, this is COOH. So, we are looking at the pK1 value of R2, the pK2 value of R1 amino acid. And what other information do we need? We have to see whether R1 and R2 themselves have any ionizable groups or not. If not, then we know that it is going to be a pK1 plus pK2 divided by 2. So, for example, if we look at allylase, this is the amino acid alanine, this is the amino acid lysine, and it has been drawn at low pH. What does it mean? It means that everything is protonated. What do we have here? This is the R1, this is the R2, and we have the amino group, that is the pK2 value is associated with this alanine, the pK1 value is associated with the lysine, and we see that one of the groups does have an ionizable side chain. So, what we do is we calculate the charges of each of these possible. How do we do that? We say that if we want to look at this, we say that the amino group here now has a charge correspond to +1. The alpha carboxylate is 0 because it still has not lost it, and the side chain is +1 from the lysine, because the alanine does not have any charge. So, what is the net charge? The net charge here is +2. If we look at now pH 5, all the values are given to here. The amino group contributed by alanine is 9.9. So, when we are at pH 5, we do not need to worry about that. The alpha carboxylate group from lysine is assigned at pK 2.2. What does it mean? It means that when I have reached pH 5, this proton is lost. So, this proton is now lost. So, the amino group will remain at +1. The carboxylate now is -1. The side chain is still +1 because we have not lost the side chain proton. So, what is the overall charge? It is +1. We go to pH 7, but do we have any changes at pH 7? No. So, it remains in the same condition where we have the same charges. We can write the same charges out and it is still +1. So, there is no change here. As soon as we come to pH 10, what happens is we lose this. So, what is this? This is the alpha amino group. So, this becomes now 0 because it has lost the proton. This remains at -1, this is +1, it is now 0. When we reach 12, we lose this. So, this becomes 0, this remains at -1, this is at 0 and this becomes -1. So, what are we interested in? We are interested in the pKa value that goes from +1 to 0. We are interested in the pKa value that takes from 0 to -1, and the average of those two is going to give us the PI of the peptide lyse. We want to go now to the next idea, and we want to consider that if we have now a greater longer rather peptide, then what do we see? So, if we have a tetrapeptide like this, we can do the same thing, tyrosine, aspartic acid, glycine. We just need to know how we are going to go about doing it. So, for an example or something that you can try, for a tetrapeptide glue lyse tyrosine, you write out the structure, consider the assigned pKa values and calculate the PI of this tetrapeptide, as you can try that out as a homework. The applications are many. We want to know for separation of proteins, for specific process called isoelectric focusing, where you can actually isolate proteins, because we look at protein migration in gel electrophoresis. Suppose we do not want the protein to migrate. If we know what the PI of the protein is, we know that the electric charge is neutral, so the protein will not migrate further. The references are, some of them are old references that I will be giving you so that you have an idea of what to read, and some of them will be relatively newer references which give you a different perspective. So, what we learned is how amino acids are amphoteric in nature, the acid base properties, the amino acids with ionizable side chains, and how we can actually determine the PI of the different peptides and further on for proteins. Thank you.